The last thing we're going to discuss today is the process known as membrane transport. And membrane transport is simply referring to how things get across a membrane, from the outside of the cell to the inside of the cell, or vice versa. It is possible for some molecules to cross a membrane between the individual phospholipids. Remember, the phospholipids are not attached to each other. They are simply attracted to each other, and that's what holds a membrane together. So it's possible for molecules to sort of squish between the phospholipids and cross the membrane. Molecules that do this, that cross membranes between the phospholipids, are using a process that we call simple diffusion. Now, you have to be a very particular type of molecule to do this. Only certain molecules can use simple diffusion to cross a membrane. Those molecules have to have three particular chemical features. First of all, they have to be relatively small. And by small, I mean they have to have a molecular weight that is less than 100. If you recall from your chemistry class, the molecular weight of a molecule is simply the atomic weights of all of the individual atoms added up. So, for example, oxygen gas, which has a chemical formula of O2. Oxygen gas has a molecular weight of 16, and that's because each one of those oxygen atoms has an atomic weight of 8. So 8 plus 8 gives the molecule a molecular weight of 16. So a molecular weight less than 100 is a relatively small molecule. That makes sense, right? You wouldn't expect something very big to be able to just cross in between those phospholipids. Now, the second characteristic that a molecule has to have to use simple diffusion is non-polar. Remember, we said for now we're going to think about that word non-polar, meaning hydrophobic, water-fearing. So a molecule needs to have that non-polar or hydrophobic quality in order to cross a membrane with simple diffusion. The last characteristic that's needed is neutrality. In other words, molecules cannot have a charge on them. If they have a charge, they won't be able to use simple diffusion to cross the membrane. So, what kind of molecules do we see passing membranes with simple diffusion? I already mentioned one, oxygen gas. Another good example would be carbon dioxide gas, CO2. If we add these atomic weights together, we get a molecular weight of 44. So these two gases can use simple diffusion to get in and out of a cell. Think about the red blood cell that's coursing through your veins right now. Red blood cells have the job of delivering oxygen to cells and removing carbon dioxide. It's very important then that the oxygen and the carbon dioxide can go in and out of cells very rapidly without any barriers. So it makes sense that they can use simple diffusion. They just sort of squish between the phospholipids and get across membranes. Now this next example, C6H12O6, this is the chemical formula for glucose. This is a big molecule. If you add all these up, 6 carbons, 12 hydrogens, 6 oxygens, you get a molecular weight of 180. So this molecule is just too big to use simple diffusion. And lastly, for an example, we see a sodium ion. That's Na, that's the symbol for sodium, with a positive charge. Any molecule that has a charge on it cannot use simple diffusion. Now, simple diffusion is an interesting process. It's a spontaneous process. And remember, we said when we use that term spontaneous, we're saying that you don't have to do anything to make it happen. You don't have to put any energy into the reaction. You don't have to use an enzyme. It just happens by itself because of chemical properties. Now, the chemical property that's going to drive simple diffusion is a concentration gradient. So, as long as the molecule that's trying to get across the membrane, as long as it meets those three chemical requirements, small, nonpolar or hydrophobic, and no charge, as long as it meets those requirements, the only other thing that needs to happen is there needs to be a concentration gradient in place. Now, what do I mean by that? By definition, a concentration gradient is simply a difference in concentration of that molecule on either side of the membrane. So, inside versus outside of the cell. Remember, all that word concentration means is the amount per volume. So for this molecule, if there's a difference in concentration between the inside and the outside of the cell, we have a gradient. We have a concentration gradient. Simple diffusion will occur when there is a concentration gradient and the molecule is small enough, hydrophobic, no charge. Now, simple diffusion will move the molecule across the membrane down its concentration gradient. Now, what the heck does that mean? That means the molecule is going to move from the side that has the higher concentration towards the side that has the lower concentration. The molecule will cross the membrane from the high side to the low side until equilibrium is reached, until there are equal concentrations on both sides of the membrane. Let's take a look at this. In this drawing, we're to read it from left to right, and we're supposed to imagine that time is passing. The artist has drawn in a depiction for a membrane, for a bilayer. This represents a cell membrane. You can see heads and heads on each side and tails in the middle. So we're to imagine that this is the inside of a cell, this area over here, and this area represents the outside of a cell, and this is the membrane that separates them. Now, we've got a molecule represented in blue. Who knows what this is? Maybe this is carbon dioxide gas. And we can see that there's a large concentration of this molecule outside the cell, and there's literally none of it inside the cell, okay? So this is an extreme example. There's a gradient here. There's a difference in concentration of our blue molecule. Now we're going to imagine that this molecule is relatively small. It has a molecular weight less than 100. It is hydrophobic, relatively speaking, and it is neutral. It has no charge on it. Now, we're going to allow a little bit of time to pass, and we're going to allow the gradient to drive simple diffusion. We're going to see the blue molecule pass from the area where the concentration is high towards the area where the concentration is low, and that's what we're seeing here. The direction of movement here is from the area where there's a high concentration across the membrane to an area where there's a low concentration. And that's going to continue across time until this happens, until there's an equal concentration on both sides of the membrane. And that is equilibrium. That will be the time that the simple diffusion process ends. Now, water, which again is critical for living cells, water can cross a membrane more than one way. But one of the ways that water crosses is with simple diffusion. Because this process is a little bit unique, because we're only talking about water here, we give this process its own name. We call this osmosis. So all osmosis is, by definition, is the simple diffusion of water across a cell membrane. Now, we're talking about water, so the terminology we've been using gets a little confusing because you don't have a concentration of water, right? Particles have a concentration, molecules have a concentration, a certain number of them per volume, but water doesn't have a concentration. So there's a difference between discussing simple diffusion in terms of molecules crossing a membrane and discussing simple diffusion in terms of water crossing the membrane. Here are the key differences I want you to be familiar with. Osmosis is going to occur when there's a concentration gradient for a molecule, but it's a molecule that cannot cross the membrane using simple diffusion. In other words, the concentration gradient can't be resolved. It can't be fixed. It can't resolve to equilibrium because you have a molecule that is either too big or polar or charged, and it can't cross the membrane. When that happens, osmosis will occur. And that's because the water will fix the concentration gradient. The water will help it go to equilibrium. So osmosis occurs when there's a concentration gradient for a molecule that can't be resolved by simple diffusion because the molecule can't cross the membrane. Osmosis will move water to get rid of that concentration gradient. And here's where it gets a little confusing. The water does not move from high to low across the membrane the way molecules do. It goes in the other direction. The water moves from the side with the low concentration of molecules towards the side with the high concentration of molecules because the water is trying to dilute the concentration. I know this is confusing. Let's take a look at an artist's depiction. On this slide, you can see a depiction of a cell, and you can see that the inside has this collection of particles. There are water molecules in here, those are shown in blue, and there's some kind of a salt molecule in here shown in white. The outside of the cell is drawn as this pinkish purple color. And of course, outside of the cell, there's also going to be water, there's also going to be salt. Imagine that we're talking about sodium chloride, and this is your body. These are cells in your body. All right, so in this drawing, you can see a concentration gradient for the salt. Remember, we don't discuss concentration gradients for water because water can't have a concentration, okay? But we have a concentration gradient for these salt molecules. I've got 1, 2, 3, 4, 5, 6 salt molecules on this side. I've got 1, 2, 3, 4 on this side. So this is a oversimplified version, but it's a gradient. There's more salt here than there is here. So I've got a gradient. Now, if this was a molecule that was small and hydrophobic and neutral, it could cross the membrane. It could go back and forth across the membrane using simple diffusion. And remember, during simple diffusion, the particles move from the side with the high concentration towards the side with the low concentration until the concentration is equal on both sides. But in this drawing, we have a problem because salt is charged. Remember, sodium chloride dissociates, it ionizes, it makes sodium with a positive charge, chloride with a negative charge. It can't cross the membrane because it's got a charge on it. So what that leaves for this cell is a concentration gradient, the white particles, that can't be resolved with simple diffusion. It can't go away with simple diffusion. What's a cell to do? You see, nature doesn't like concentration gradients. Nature likes equilibrium. So there's something happening here that nature would like to fix. But the particles can't move through simple diffusion. What's going to happen next is osmosis. The particles can't cross the membrane with simple diffusion, but the water can. So what's going to happen is the water will move not from the high side of salt to the low side of salt, but exactly the opposite. The water will move from the side where there's less salt towards the side where there's more salt. Think of osmosis as diluting out the particles. By bringing more water into this cell, I'm going to change the concentration of the salt. Remember, concentration means the amount of salt per volume. So if I increase the volume of water in here, I'm going to get rid of the gradient of the salt. That's what osmosis does. Osmosis only occurs in living cells when there's a gradient of a molecule that can't go away. It can't be fixed because the molecule can't cross the membrane. What if we have a molecule that can't use simple diffusion, but the cell needs to get it across the membrane? It's not enough just to get rid of the gradient. It's not enough for osmosis to get rid of the gradient. What happens if a cell needs to bring a molecule in or get a molecule out, but it's too big, or it's too polar, or it has a charge on it? Well, that's where the other membrane transport mechanisms come in, and there are two of them that we need to be familiar with. So any molecule that cannot cross a membrane using simple diffusion has to use membrane proteins to get the job done. And there are two protein-assisted processes. The first one is called facilitated diffusion, and the second one is called active transport. Facilitated diffusion has a perfect name. It has a name that describes this process. It's another type of diffusion. It's going to be similar to simple diffusion in some ways. What makes it different is that it's facilitated or assisted with protein. We're going to see molecules moving down the concentration gradient, just like we do with simple diffusion. The molecules are going to move across the membrane from the side with the high concentration to the side with the low concentration. It's going to be spontaneous. We're not going to need to add any energy to make this happen, but it's going to happen with the assistance of a protein. Instead of the molecule crossing between the phospholipids, the molecule is going to be helped across the membrane with a protein. That's the difference between simple diffusion and facilitated diffusion. Now, active transport is different. Active transport is different from both simple and facilitated diffusion. The primary difference is this. Active transport is not spontaneous. Active transport requires energy. And the reason it requires energy is this. Active transport is going to move molecules across a membrane with the assistance of a protein up their concentration gradient, not down. So active transport is going to move molecules from the side with the low concentration towards the side with the high concentration. Another way to think of that is active transport is going to maintain a concentration gradient. It's not going to resolve it. It's going to maintain it. That requires energy, and that requires the assistance of a transport protein. We give these transport proteins a name. We call them pumps. Facilitated diffusion requires proteins. Active transport requires proteins. Facilitated diffusion involves two different types of proteins, depending on the molecule that's being carried across the membrane. Some of these are called carrier proteins. They are literally going to carry the molecule across the membrane. And the other ones are called channel proteins. Channel proteins have literally an open channel in the middle of the protein. They have a tunnel through them. And the molecule that's trying to cross the membrane is just going to pass through that tunnel. Carrier proteins are different. Carriers are going to bind to the molecule that's trying to come across, and then they're literally going to change shape and pass that molecule over. So carrier proteins really are physically carrying the molecule across the membrane by changing their shape. And channel proteins are letting the molecule pass through the tunnel in the middle of them. This drawing over here represents a pump protein, and they haven't drawn any particular features of this protein. The key thing that they've drawn is this. They've drawn this ATP molecule, this little energy powerhouse molecule. We're going to need the ATP to give us the energy to do active transport. And the pump protein is going to work to maintain the gradient. See how there's more of this molecule shown in blue? There's more of it outside of the cell than there is inside of this cell. Here's the membrane. What the pump is going to do is not move it from the high side to the low side. No, the pump is going to do the opposite. It's going to take it from the low side and move it to the high side. It's going to keep that gradient in place. And yes, there are times during the life of a living cell where certain gradients have to be maintained in order for certain cellular processes to occur. The last two slides for this lecture are just a look at the ribosome. Remember we said all living cells contain ribosomes? I just want to introduce them today. There's a drawing over here of what a ribosome looks like, and this is actually pretty accurate. This blue line here, this represents a messenger RNA molecule. We haven't talked about nucleic acid yet, but DNA and RNA are the two forms of nucleic acid that we find in living cells. And ribosomes, remember, have the job of making protein by using the code that's found in messenger RNA. This ribosome, the way it's drawn, has two subunits, and all ribosomes have two subunits in them. They have a large subunit and a small subunit. Both subunits of a ribosome contain protein and another type of RNA called rRNA. That just stands for ribosomal RNA. Ribosomes are making protein. They're performing a process called translation. Translation is the process where a messenger RNA molecule is being used as the code to make proteins. Now, all cells have ribosomes, but there are some structural differences between prokaryotic ribosomes and eukaryotic ribosomes. All kinds have a large subunit and a small subunit, but the actual protein that's inside those subunits, the actual rRNA that's inside those subunits, is going to be a little bit different in bacteria and archaea than it is in eukarya. So yes, when we look at these drawings, we can see these little dots that represent ribosomes. All cells have ribosomes. This prokaryotic cell that has no organelles in it, it's got ribosomes. This eukaryotic cell, this animal cell, has ribosomes in it, the little blue dots. This plant cell, eukaryotic cell, has ribosomes in it, little blue dots. All living cells have ribosomes because ribosomes are not organelles. Remember, organelles have to have membrane around them, and ribosomes do not have any membrane.